So, good evening today. Thanks for daily filling out the daily reflection form. I could track your comments, and I'm very happy that many of you find it very positive and you are learning very vibrantly. So, it's happy to see, and many of your results are also very much productive. I will do one more video on commenting on your results, but most of the time, I see most of the questions regarding constructing the phylogram or Excel or deriving this pairwise genetic distance data. Their answer is already there in the video. Most of the time, maybe I don't know how it is being missed, but the instructions are very clear in video and it can be make use of. So today we thought that we will do a frequently asked questions in barcoding. So this video. So regarding sequencing update, we've been sending at least 5 results every day. It's been dispersed. Once you get your sequences in your inbox, please use them for blast analysis, phylogeny, and also pairwise distance data. That's the intention behind it. From next week onwards, we will be getting into formulating your manuscripts. Okay, whichever we are planning, we will see how to make a manuscript in barcoding. You don't need to have your own data for that. Even barcode gap analysis can be something we can think about that can be done in 5 days using pairwise distance data. If you can see, we can propose some new barcode gaps. That will be much more acceptable in editor's point of view. So today I'm reading your questions. I thought I will do a question answer session. So there is a question, as you have said, there are diversified species in India. There is no evidence of the species. How can we explore it? Sir, ma'am, you need to know that we have something called Zoo Taxa, the journal which publishes monographs, list of plant species, animal species. It is because of listed species per country. We know if there is something new species, then could be an alien or invasive. That's how they all have a clear-cut depository listing out every species in ecosystems like coastal wetlands, mangrove ecosystems, lagoons. For animals and plants, there are many publications something like this. World checklist of vascular plants, checklist of polychaetes, checklist of both nematodes, bivalves, even fungi, mushrooms. You need to check for checklist if you are working on, say, for example, mushrooms. Look for checklist. Checklist is a research article publishing list of species in a particular ecosystem. So Zoological Survey of India, Botanical Survey of India also publishes catalogs, animal discoveries, something like this. You can use this to find list of species available around us. So these are the ways to look. I hope you will take a look. So provide career opportunity related to this summer internship. So each students can apply their knowledge in real world. Yes, of course, whatever you are doing right now is actually an industrial requirement. Here is a proof. Okay. In Bangalore, I know many of the sequencing companies need people in analyzing chromatograms, extracting sequences, do blast analysis, build a phylogram, prepare a species identification report and send them to the clients. They don't have much hands in this area. That's why we, instead of training the people for 6 months and hiring them, that will run an internship. Even barcode biotechnology requires such a people. Still, we are getting CVs and we are not finding the right people. And this is a Eurofins and other big sequencing players require people in this skill set. Okay, that's why this internship is happening. I hope you can simply browse and visit these websites, see who is a dominant player in the DNA sequencing, and you can get the answer from there. There are many advertisements circulated like this. And I want to know sequences that have been provided in assignment to whether the sequence is coming from plasmid DNA or genomic DNA. As I said, the assignment to, I guess it is regarding to CO1 sequences. So those come from mitochondrial DNA. If it is RBCL or METK, they come from chloroplast genome. If it is a fungal, it is from ITS region. Okay. So bacteria, it is 16S RNA. So it's neither nuclear or plasmid because it's an animal sequence. So it comes from mitochondrial genome, cytochrome C oxidase subunit 1. I would learn about, see, protein plus. Yes, we will do one more session. This session probably on Wednesday we will have, because once you get your sequences, there will be a session on how to submit your sequence to GenBank and publish it and get the accession number, for which, to prove that our protein coding genes don't have gaps or stop codons inside the amino acid sequence or inside the DNA sequence, We will translate them into amino acid. We will do the protein blast in that session. It will be included. See how you load the sequence. So wet lab work. Okay, we already have more people inside the lab working at labs. We can arrange a demo if possible, or we can access the sequencer room CCTV. I can do one more session on that. Let's see how to go about it. But there are lots of videos available in YouTube for Sanger sequencing, for Illumina, HiSeq, MiSeq, even Nova sequencing. Oxford Nanopost, many videos are available in YouTube. I would encourage to check that. If necessary, we will do something about this. So there is a big question. Okay, I don't know how the Zoom got disconnected. So protocols for DNA extraction, protocol optimization, degraded sample. Yes, we are doing. Even I told you we are having a forensic level, high precise DNA reagents. Even degraded sample, we are extracting DNA. Comparison between Sanger's and next-gen sequencing, use of automated pipelines like KIME2, OVI tools, Moto for meta barcoding, phylogenetic tree building using ASAP methods and handling large data. As you know, this is not a big data Illumina next-gen sequence analysis internship. We are concerning or we are just focusing on Sanger sequencing. As you know, the company has a 1,000 barcode initiative where we are encouraging local species yet to be barcoded, which can be done only through Sanger sequencing. That's why this internship. We also used to run internship in next-gen sequence analysis where all these packages are included there. Probably next month we will have, at the end of the month, we may have a next-gen sequence analysis platforms during which we will discuss all those things. But right now, this is not our objective. So I'm a taxonomist. How can I like to know more about how we can be a part of this program from where we can get funds? See, for funding, we can have a state agencies, central agencies. We also have a corporate responsibility, social responsibility funds from corporates. We used to get these funds, but I will tell you the basic qualification of a principal investigator to apply for funds is to have a publication. No one is going to give us money if we don't show them how to what is our achievement, how we can use the money to produce a paper. Okay, so that's why this internship, if you have selected a first time barcoded species, we are going to write a manuscript and publish it, use that as a proof and write a research proposal, and we can see how to secure the funds. There are a lot of sources. We'll see once we have a manuscript published. How to set up a small lab for UG and PG students. Okay, we also have something called Barcode Academy. We help government schools by providing free actually DNA isolation reagents for plant DNA extraction. Other using simple reagents, we can do that. We are doing actually, and we can also do a hands-on demonstration. We used to carry our gel electrophoresis apparatus, everything to the school, and we run a workshops there. These are all given for free. But if you want to establish a lab, I would say try to get small funds from the state government. We have a KSRTC fund, TNSTC fund, Gujarat government biotech funds. Small funds we can try to get and see how to establish a lab. Or I would say instead of learning the bioinformatics skill and seeking for a job, better start a startup. Few friends and colleagues coming together, start a startup. You can attract a lot of seed funds from initial stages of startups. Startups these days don't even need to have a physical space. It can be done virtually also. Virtual company and virtual startup. You can attract funds for bioinformatics analysis. You can sign a contract with sequencing companies. You can get the data from them, analyze it, and prepare a species identification report, mail them back, or mail to their customer. You can be a consultant to the startup you have started, can be a consultant. We have nearly 17, we have signed MOU with the 17 startups in barcode biotechnology. We are using them to analyze sequences. Okay, so I think bioinformatics UGPG students can come together and they can initially start a startup, easily start a startup. Sorry. And this can be very much useful. So yes, more about seaweed barcoding. I will encourage you to read more articles that are coming seaweed barcoding. There are many good articles they've been published very recently. They are using eDNA, metadNA, and also DNA barcoding. As I said, in my estimate, at least still 7 to 8 species of that is native to India are yet to be barcoded from the coastal waters of India. So you can find the species name through the checklist, check for the sequences in the NCBI, and you will get it, which are barcode or which are not. I hope this session is useful since we are into phylogram construction and pairwise distance data analysis. So today there will not be any live news session. Let's focus more on completing these all four tasks and let's finish the task before today. From tomorrow onwards, we will be focusing on how to submit the sequences and get the accession number and how to write a manuscript about it. Thank you.